My name is Elena Moreno. I'm the project manager for HIV prevention, research and development at IAS. It's my pleasure to open this first webinar in a panel discussion in a series of experimental medicine for HIV preventative vaccines. Today is the first webinar that is entitled "What is experimental medicine and what are the key challenges?" I would like to welcome all participants and our panelists who will be introduced to you shortly. Let me just share a few rules for the webinar. Note that all participants are going to be muted. If you have any questions, you can use the questions and answer box that is on the bottom of your screen. Please remember to focus on the questions that are related to the webinar. If you have further questions, you can always send us through our email at this enterprise@iassociety.org. If you have any technical questions, you can use the chat. If there is any technical issue, you can use the chat. So thanks. I'm now handing over to Linda Gelbecker, who will introduce today's webinar. Thanks, Linda.
Thank you so much, Elena, and a real pleasure to welcome you all to this online seminar. On behalf of the Global Vaccine Enterprise and the International AIDS Society, my name is Linda Gelbecker. I come from the Desmond Tutu HIV Center here at the University of Cape Town, all the way down at the bottom of Africa. But this online seminar is the first in our series entitled, "What is Experimental Medicine and What are the Key Challenges?" And you may have an idea of what experimental medicine is, or indeed you may be completely new to this topic. We would ask you to hold on to your hat and stay engaged, because we are going to assume that all will be learning about this, but very quickly over a two webinar series, we hope we will be bringing everybody up to speed on this new concept. Today, we are going to be defining what experimental medicine is within the context of HIV vaccine research and discuss the issues relating to the importance of these kinds of studies, what the added benefits of experimental medicine research brings, how we engage our communities, very importantly, to actually become involved and provide clear communication, ethical considerations of the research, and the implications of these trials in the context of both industry, researchers, and regulators. It's my pleasure today to introduce our panelists and then hand over also in a moment to the moderator who will be managing the discussion part of today's session. Let me work through the panel. First of all, Professor Robin Shattock, who is the head of mucosal infection and immunity within the Department of Medicine at Imperial College London. And of course, Robin is very well known to us in the HIV field and increasingly an important figure in the world of experimental medicine. Dr. Christine Grady is a bioethicist and senior investigator who serves as the chief of the Department of Bioethics at the US National Institutes of Health Clinical Center. And again, very important expertise and understanding on the ethics of this kind of research. Dr. Jerome Kim is very well known to us again in the world of vaccines and is the Director General at the International Vaccine Institute. And we certainly welcome Jerome to share some of his ideas and understanding of this topic. Maureen Luba, a good friend of ours, you will have heard her voice on numerous occasions on podcasts and in webinars. She is a senior program manager at AVAC and is working in global advocacy for HIV prevention and a vaccine champion. And then finally, very happy to welcome Carl Dieffenbach. Dr. Dieffenbach serves as the director of the Division of AIDS at the National Institutes of Health, and obviously a very important role in vaccine development in the field of HIV. I am now happy to hand over to Professor Robin Shattock, who will be providing a presentation introducing us all to experimental medicine before we continue with the panel discussion Q&A, as I say, hosted by Dr. Mark Feinberg. So over to you, Robin.
Thank you very much, and welcome everybody. Today I'm going to talk to you about experimental medicine trials, and you'll see I've inserted vaccine into the title. And the reason for that is if anybody goes to PubMed and puts in experimental medicine trial, you get over 100,000 hits. And obviously any human study is in its essence experimental by nature. A few disclaimers. First of all, the views and opinions that I'll express do not reflect official policy or a position of any organization. And my intention is to stimulate discussion rather than providing a definitive or comprehensive position. Well, at its very basic level, experimental medicine trials have been performed to further scientific understanding of vaccination and human immunology for several decades. So this is not a new concept. However, most of this previous research has been done through conventional pipelines. And I'll give you a couple of quick examples. So the first would be the use of licensed vaccines in terms of studying human immunology. And just two recent examples of many hundreds would be a paper, a nice publication looking at PET-CT imaging to look at lymph node activation using 2 licensed human papillomavirus vaccines. A small study, 27 participants, but used in adult women who were likely to get limited benefit from the vaccination itself, given that the target age for vaccination with the HPV vaccine is in adolescent before the onset of sexual participation. And a second example, a study using a licensed influenza vaccine, looking at how germinal centers are engage memory and naive B cells. A very small study, 8 participants, and likely to benefit those participants in terms of protection against influenza. And I think all of us are familiar with using licensed vaccines in this type of immunological research. The second, which is also, I expect, very familiar to people, is applying in-depth immunological analysis alongside product development programs using experimental vaccines. And I've given 2 examples there, taken from SARS-CoV-2 vaccines, and obviously there will be an explosion of publications that have been immunological studies allied to product development programs. These are uncontroversial, but obviously show how experimental medicine studies can be used in conventional contexts and have been used for many decades. But I want us, for the sake of today, to have a more precise or more defined definition of experimental vaccine trials. And here my definition would be clinical investigations undertaken to test or generate scientific hypotheses that advance vaccine discovery and development, but provide no direct prophylactic or therapeutic benefit to the participant. So in effect, this is undertaking scientific discovery using vaccines that allow us to test specific scientific hypotheses, but in their own right are unlikely to provide benefit to the participant. These differ from product development trials, where plausible candidates are positioned to progress through conventional phase 1 to 4 testing. While they're often conducted under phase 1 guidelines, the focus of experimental medicine vaccine trials is to accelerate vaccine science rather than to progress individual products. Often they are iterative in nature, and there is little or no expectation that these will progress further in their own right. Now, one of the common misconceptions that I think it's important to exclude from the start is while the aims of experimental vaccine trials are similar, these are not phase 0 trials. Phase 0 trials are specifically deal with microdosing studies of therapeutic drugs. They were designed by the FDA to accelerate and de-risk drug development and were implemented in response to high levels of late-stage failure of therapeutic drugs. Now, phase 0 trials in the US can use exploratory INDs, but the legislation specifically excludes vaccines. Now, you can see that there are some similar urgent needs in the vaccine field in terms of de-risking vaccine development and implementing an approach to reduce high levels of late-stage failure, particularly in terms of HIV vaccine design. So this perhaps represents a gap currently in legislation for vaccine research and development, particularly with respect to the FDA. So the definition clinical investigations to undertake or test, to test or generate scientific hypotheses that advance vaccine development is clear. But what perhaps is requires more consideration is the fact that they provide no direct prophylactic or therapeutic benefit to the participant. So that raises the question, why would you even want to conduct such types of approach? Well, here are the whys, or some of the whys, for the use of experimental medicine vaccine trials. Scientifically, we know that animal models are often not fully predictive of human responses. For example, there are germline differences between humans and experimental animals, which means that often many principles cannot really be effectively determined unless they're performed in humans. And so there is an increasing recognition of a need for human immunogen discovery based on human trials and using in-depth, state-of-the-art immunological analysis. Ultimately, the best model for a human is a human. Financially, there are also good arguments that essentially it's really unsustainable to continue to put wishful candidates into expensive phase 3 trials, and better allocations of resources are needed to increase the likelihood of success. So essentially to test things early, eliminate those things that are unlikely to succeed, and reduce the risk of late-stage failure. Logistically, we are all very aware of the public health need to accelerate HIV vaccine development and increasing difficulty in conducting large-scale efficacy trials, where the environment is becoming much more complex with an increasing range of other preventative measures that reduce incidence of HIV and make the job of testing for efficacy increasingly high. And then ethically, I think increasingly enrolling volunteers in vaccine trials with a higher probability of success is an important consideration and focus. Reducing the number of volunteers exposed to unsuccessful vaccines is also important. But of course, using experimental vaccine trials requires clear communication around the lack of benefit to the participant. So what sort of things can be used with an EMVT approach? Obviously looking at accelerating HIV vaccine development and increasing the success of products that then move into clinical evaluation. Importantly, they can be used to address questions that are not capable of definitive solutions solely in animal models. They provide clear opportunities for early iterations between preclinical and clinical investigation. So in their own right, they also improve preclinical models and their predictive utility. They can be used for early validation and sequential iterations of structural-based designs. And I think we increasingly understand that structural-based design may be one of the important pillars for realizing an effective preventative HIV vaccine. They can be useful for evaluating novel concepts prior to formal product development. And they allow very intensive, in-depth human analysis to a level that might not be applicable to a conventional phase 1 study design. So this table really highlights the difference between a traditional phase 1 and an experimental phase 1. And by and large, most experimental medicine trials are still conducted under phase 1 designs. What you can see is that the focus is different, although many of the components are similar. So let me give you a quick example of what experimental vaccine trial might be. One of the very current examples would be clinical evaluation of a germline targeting immunogen. So for example, the EOD-GT8 germline targeting construct developed by Bill Sheath that is currently in clinical evaluation. Here, this tests the scientific concept that a certain immunogen can stimulate relevant germline B cells in a significant number of individuals. And while this is a very critical, important scientific question, the induced antibodies are likely to provide no or very unlikely to provide any benefit to the participant. So this is a very much experimental medicine question that is important to answer, but won't directly benefit those participants. I'll give you a second example, and this is the use of native-like trimers as challenge agents to further inform our understandings of the pathways to induce neutralizing antibody breadth. This is an example taken from the European AIDS Vaccine Initiative program. Here, a range over 8 different trimers was supported by a single pivotal toxicology package. There was a use of phase-appropriate small batch manufacturing that reduced costs by a significant margin. It was highly exploratory, so hypothesis generating. But we recognized the challenge immunogens were unlikely to provide any meaningful protection in their own right. This approach provides significant savings in time and cost, accelerating into the use of native-like trimers. In this example, this was allowed to be performed not as a phase 1 study with regulatory oversight, but as an experimental medicine trial with only ethical oversight, an approach that is applicable in the UK, but not currently in the US. And I think both of these examples recognize that we are now in a situation where we are likely to need a large number of iterative experimental medicine vaccine trials to identify the series of immunogens or are able to induce broadly neutralizing antibody responses before a realistic product is identified. Now, that is a bit of a step change, both in terms of funders and researchers, to actually stand up and recognize that a lot of scientific research is needed in clinical trials rather than having to justify that every clinical trial is part of a product development program. So what are the potential savings in terms of cost and money? Well, savings and time costs can be gained from adopting a generic manufacturing process, what we call platform approaches, whether it's for proteins, vectors, RNA, or any other approach. Also doing smaller scale batch production, phase appropriate, can reduce costs and save in terms of time. Adoption of generic release and stability criteria can get products through into clinical trials faster. Acceptance of generic toxicology for multiple products, again, will allow a faster transition to clinical investigation, and then savings through parallel rather than sequential product development programs. So streamlined product development can achieve and support iterative experimental medicine vaccine trials. And certainly here, small experienced teams can often provide cost-effective management across platform approaches with rapid decision-making and flexibility to address scientific priorities during a clinical program. What are the potential challenges? Well, there are many challenges from a regulatory position. Certainly thinking of the US context, while the FDA have special provisions to de-risk drug development and late-stage failures, those provisions currently do not exist formally for vaccine development. The caveat there is obviously the FDA are very open to pre-IND discussions and showing flexibility around approaches. Investigator-initiated studies in the US may provide some flexibility around using research INDs, which are different to commercial INDs, and trial design at the phase 1 level, where reduced specification in the trial design can be scientifically justified. Now, some of the challenges are different regulatory bodies can provide greater flexibility over the regulation of experimental medicine trials. And these seem to be defined by and large at a national level. That means that there is no global standard. And so when we think about conducting experimental vaccine trials in many different parts of the world, particularly parts of the world where there may be less mature regulatory authorities, it's a challenge where there's no common standard. Often interpretation of whether something needs to be done at phase 1 or through a non-phase 1 approach is down to the definition of a product. So some agencies define a product as an agent that has therapeutic prophylactic activity. And so something that is scientifically argued not to provide benefit to the participant falls out of the remit of a clinical product. Whereas other agencies, particularly the FDA, define a product as any substance or biologic that has any physiological effect. So for example, the FDA would define a human challenge agent to be used in a human challenge trial as a product, even though that clearly doesn't provide a therapeutic or prophylactic benefit to the participant. Platform approvals for groups of immunogens is still relatively novel for a number of regulators and something that needs to be built on. And obviously there is clear need for more regulatory discussion and debate, particularly in the context of HIV, where the risk of success in late-stage trials needs to be improved by a higher throughput of early discovery-based trials. The other potential challenges are financial. If experimental medicine trials do need to be performed along classical phase 1 designs, there may be little savings in time or money. So therefore, one needs to potentially look for alternative pathways or better discussion with regulators to make those time and cost savings. Logistically, phase-appropriate manufacturing is often not commercially attractive, and so it's often hard to negotiate costs for small batch manufacture, although I suspect that will change with some of the new technologies like mRNA production. And then ethically, obviously, there are many challenges in terms of communication of the lack of benefit, where volunteers are essentially seen as research partners in solving scientific questions and improving vaccine discovery rather than getting a vaccine that will benefit them. And that becomes more complex as we think about vulnerable groups or those with a low educational background in terms of vaccine discovery and human trials. And finally, the lack of benefit may also argue for a further need for greater incentivization for people to participate in these types of trials. So I hope I've given you a good introduction to think about the space that experimental medicine vaccine trials could have in HIV discovery. I think potentially there is a lot to gain from adopting experimental medicine type trials to down select and enhance our understanding of what it takes to develop protective and therapeutic vaccines before they move into a conventional product development program. I think we're all aware of an urgent need to change the paradigm for HIV vaccine research. And by doing this and being active in this area, it has the potential to drive innovative use of experimental vaccine trials that will provide wider benefit to vaccinology for many other unmet needs. And I will stop there and hand over to Mark to chair and stimulate the discussion.
Thanks, Robin. I'll intervene just quickly to say, first of all, thank you very much to you. I think that has laid an extraordinary, broad and clear framework for us to start our discussions. But I want to just quickly introduce Mark. Real pleasure to have you on the webinar this afternoon. Dr. Mark Feinberg, of course, the director of IAVI, and an organization that for many years has not only led the way in terms of vaccine research, but also more recently the way in the application of experimental medicine type trials in forging a new paradigm for HIV vaccine research. And with that, Mark, I want to hand over to you and ask you to take the panel discussion forward so we can deepen our understanding of this. Thank you again, Robin, for a great talk. Over to you, Mark.
No, thanks very much, Linda Gale, and for your introduction and for the invitation to participate. And thanks, Robin, for a great framing discussion. We have a series of questions that we want to discuss with the panel that I think Robin laid a very good groundwork for, but we definitely want all of the audience members to put questions in the Q&A function as well, and we'll do our best to work through as many of them as we can. As you heard earlier from Linda Gale, we have not only a great speaker today, but we have a great group of panelists who can address a wide range of questions related to the concept and the practical applications of the concept of experimental medicine studies. So I can ask the presenters all to put their video on the panelists. That would be great. And maybe I can start this question by asking Carl about how he sees the importance of experimental medicine studies in accelerating HIV R&D activities and why they're getting such attention now. Clearly with the NIH being the largest funder of HIV vaccine research and having been in that capacity for many, many years. Carl, you have a very unique perspective in looking across the landscape and trying to understand how the different components of vaccine development and clinical evaluation fit together. And I'm interested in hearing your perspectives about the importance of experimental medicine studies at this stage of the HIV vaccine research endeavor.
Thank you. And I really appreciate the opportunity to participate in this webinar today. I think this is an incredibly well-timed event in terms of helping the community of people interested in HIV vaccines consider where we are and where we need to get to over the next period of time to get us to a safe, effective, and durable HIV vaccine. We have tried an approach for the last decades that was really been based on more clinical development models, although some of the vaccines have had a fair amount of discovery associated with them. I think it really is time for us to consider an approach where we start from a set of building blocks. And the advantage of an experimental medicine approach is you can test very specific questions with appropriate design in a safe and ethical way so that you can then define in a very precise way how you are building your vaccine concept. The two examples that Robin gave are excellent in terms of helping to explain how an experimental medicine trial will advance the field. So I think it is an essential component that we need to incorporate into our consideration of how we advance these. There's a couple of handoffs that we also need to be aware of. So if we're starting from the position of material that is manufactured in a research laboratory, there's no way a single research laboratory can ever take a vaccine much beyond experimental medicine or phase 1 into development. So there has to be some sort of full scope of development that is considered at some point in this process. We can't think just about this in a relatively linear fashion, that A to B to C to D. We have to think about the end game and how we get from where we are today to something that is conceivably a product. I realize that's a daunting task, but this is where our challenge is today. We have had virtually no success really in the HIV vaccine field. So we have to fundamentally approach this differently in terms of getting immune responses that are significantly better than the response that the body can make directed against a natural immunogen. And that's where I think the EOD GT8 is such a breakthrough, as a particle approach that is a very specific target. And then the experiment was done to determine the level of response. My concern is a little bit about the degree of forward motion. Experimental medicine studies should be able to be designed and performed and get an answer. If every single experimental medicine trial ends up lasting a year or a year and a half, it's going to take us decades to get to a safe, effective, and durable HIV vaccine. So there needs to be rigor in what we test, how we test it, and then the process becoming much more straightforward and simplified, because what we're seeking is immune response. We have to continually focus on the ethics of this question. We cannot be seen as using study participants without some sort of consultation, continued community engagement. That's going to be one of our biggest challenges. So I think it is an incredibly important activity. We need to figure out how to bring scientific rigor to selecting the immunogens that go forward. I would much prefer to be in a situation of testing hypotheses rather than hypothesis generation. That, to me, gets into a little bit of a slippery slope. So I will stop there and say I'm all for it. The NIH is very interested in exploring ways to make this a reality.
Thanks very much, Carl. You raised a lot of important points there that we'll come back to later in the discussion, including the key issues of ethics, community engagement, and accurate communication. I wonder if I might broaden the circle a little bit and bring in perhaps Jerome and Robin to sort of have them share their perspectives about the same question around how do they see this contributing to HIV vaccine development now, and what are some of the key kinds of questions that we will potentially have the opportunity to address using experimental medicine approaches.
I'm happy to start. I mean, I think, like Carl, we recognize that business as usual is no longer acceptable. We need to accelerate vaccine discovery, and for HIV, that really needs to be an acceleration of human trials. But at the same time, that doesn't mean compromising on safety. We're all still very familiar with the huge number of questions over the safety of COVID vaccines, given that they were developed in such a short space of time. And so it's still important for us to make sure that we take the public with us and do due diligence on the safety while accelerating the progress of vaccines into the clinic in relatively quick time, iteratively, in order to really make those very needed very needed cuts, almost paradigm changes to the acceleration of vaccine research.
Thanks, Robin. Jerome, any additional thoughts?
Thanks, Mark. I think I'd like to skip to the end of what Carl was saying about the end-to-end thinking. You know, a part of acceleration of product development is not only thinking about having a great product, but thinking through the process and then thinking through who's going to manufacture it and efficient testing. And I think that you could actually, with experimental medicine, unless it's well thought through, generate so many different products that you won't have an efficient mechanism to actually get to the thing that you want in the end, which is an HIV vaccine. And so there's the element of timeliness, of timing and delivery is really critical. And we really have to, you know, and again, so IAVI doesn't work on HIV vaccines, we work on others. And we certainly have been in situations where products that looked interesting just didn't work out because when we tried to manufacture them, it turns out that the manufacturing steps in the laboratory did not correspond to the manufacturing steps at a manufacturer. And so the process development piece is something that we find very critical. Now, having a platform may get you around that issue to some extent, but in the end, the process is the product, as they always say. And unless you can have and you know, an organization maybe like NIH or the Horizons program could actually probably set up a process that will move things through a very orderly fashion to ensure that whatever is tested in experimental medicine trials actually has a shot at being scalable and manufacturable. And then the other piece is, you know, finding a company or somebody who's going to make it in sufficient quantities in order to do the appropriate testing as you move downstream. So I think experimental medicine is really critical. And you know, we've used it in my old program, an HIV vaccine research program in the military, to do a lot of work on chronically on cure studies. And you can use the same things like lymph node biopsies and mucosal biopsies, bone marrow biopsies. You know, you can do lots of things, but you have to focus the questions well enough so that they actually do incrementally advance not only the science, but the vaccine.
Thanks, Jerome. Well, all of you have, you know, sort of emphasized the importance of having, you know, clear hypotheses and a well-developed vision about how the experimental medicine studies might either connect to or not, you know, product development activities, but certainly providing key insights that would enable product development activities even when they don't directly test product candidates. And that speaks to, you know, I think what, you know, Carl had highlighted as a different, you know, kind of thinking that needs to be installed in the HIV vaccine field at this stage. You know, there are, you know, key questions that we know need to be answered. And given that experimental medicine studies can be very informative if they're very well designed and executed, but may not add value if they're not, and given their nature, and as we'll talk about, the issues related to important issues around both regulatory science as well as human subjects protection, I wanted to follow up on something that Carl alluded to but didn't state explicitly, is the potential value of a more coordinated approach where there's a vision of across the field where there could be more alignment so that the key outstanding questions can be addressed in the most efficient and effective way. And I think that's something that, you know, Carl's perspectives would be really helpful. And I'm sure Robin and Jerome and probably others have thoughts about that as well.
Thanks, Mark. I think we all really feel this need. We have seen cases in vaccine field and other fields where there isn't coordination, where people will, or individual groups will have funding to head down a development path even though there's a general consensus across the field that some approaches are more likely to succeed than others, but because of fundings and pressures, and frankly, a little bit of nationalism, things move forward. And it's unfortunate because that wastes time and effort. And it also burns credibility with our community that we need to be working hand in glove with. So within the NIH and within the US, we are seeking to create a system where the major players, the Gates Foundation, IAVI, the NIH-funded CHAVD groups, and anybody with a vaccine concept as an R01 investigator or others can come together and share information. I think the first is creating a platform where there's open and transparent communication about what people are thinking and what they're doing, and what is then the evidence that's available. Second is the sharing of models. When we talk about hypothesis generation, if you think a vaccine immunogen is going to trigger a given response, you can genetically construct now relatively easily mouse models with those target elements, and then demonstrate that you can highly specifically activate that specific B cell lineage in vitro, in vivo, in vitro, in the furry test tube before you ever consider putting it in people. So in that sense, you have taken the step forward on safety and also then some level of assuming and assuring that you will at least have a better than reasonable chance of having success when you put said immunogen in people. So I think there are things that we could do in terms of sharing models, sharing information as a starter, so that we eliminate duplication and communicate not just our successes, but our failures. I think that the demonstration of this given vaccine plus this adjuvant in this population or in this animal model led to essentially antibodies that were not effective or against an irrelevant part of the immunogen really helps us to focus. It's also about using the tools we have now to evaluate immunogens. It's not just about ELISA titer anymore. It's in many ways the kind of thing that Andrew Ward has been doing with his electron micrograph that looks at exactly where the antibodies are binding in an effective way, or at the bottom of the trimer where nobody needs to be making an immune response. So that's a long-winded answer, but I think we need more of those things: communication, sharing data, both positive and negative, and also then ultimately reagents and animal models, so that we can build not just within the United States, but hopefully with the European colleagues and around the world.
No, thanks for that very helpful perspective, Carl. And many people, including many potential participants on this call, may not actually appreciate how many of the ingredients for the kind of vision you're talking about are actually in place now, and there's an opportunity to bring them together in a more powerful way. But I mean, I think people look at the HIV vaccine field and compare it to the COVID vaccine effort and think that we haven't made any progress in HIV vaccine development. But all of us who are involved in it know that that's not the case. And in fact, there are really promising both partnerships as well as technologies that can really, I believe, help us move more quickly. And I'm interested in hearing, Jerome and Robin's thoughts about that issue as well, about the benefits of the issues around, thoughtful communication, clear collaboration opportunities, and a kind of more a vision to test hypotheses across the field.
I mean, I think that communication is absolutely key to solving these issues. I think that the HIV field actually has matured and is pretty good at communication, and we shouldn't forget that the enterprise is one of those bodies that's role is to coordinate the different aspects of research and be a space for us to discuss issues like we are today. But clearly, this issue cannot be solved by an individual research group. And I think increasingly everybody is looking to work together, and also looking to have validation of their experiments, their assays, their reagents, their samples by other groups. And I think now there's an increasing need actually for verification and reproducibility, which can only be provided by people doing the same things in different parts of the world.
So can I just pose a question to Robin and Carl? So is there a possibility that so having reagents made in small batches by small contract manufacturers could actually delay evaluation or cause a negative evaluation of a product that was not correctly made, not properly, I mean, when we do a product, as we're developing the process, there are dozens, maybe tens, hundreds of different analytical assays that are applied to the product through the process of purification. If those are not done correctly and you don't have the right product, it could actually bias. And how do you ensure that those, I mean, you would have a superstructure, some other kind of industrial secretariat that would ensure that the products that were going forward into experimental medicine had the right qualities, so that if any one of them looks promising or makes a significant incremental improvement, they can be moved into, say, true phase 1 testing. Otherwise, you could actually, go into one of these cycles where you're, you think that something's working or you think that something's not working, and in fact, it was the product. So I think that's one question. The second question is, I think I think Robin, you mentioned this also, that this isn't necessarily acceleration of vaccine development, but it's actually allowing us to do what we didn't have the time to do during COVID, which is to think through the big questions, because HIV is not a typical pathogen, unlike COVID in general. And it's 30 years of work has not moved us as far as we want to be. And actually 40 years of work. What am I thinking? And so, we are going to have to rethink this, and understanding the things that we understand now, many of which could actually probably be back applied to COVID as we tried to think through the tough problems of transmission, like as we do for HIV. I think that these are all things that are going to be reinforcing. I think the one last question I have for Robin and Carl is, how do we avoid groupthink around this? Because this is going to require a degree of coordination and a degree of agreement on certain things. How do we ensure that a novel idea is not going to be, suppressed by the majority?
I'm happy to have a quick start at that. So in terms of your questions about, the quality of what's being produced, obviously those are critical, and the highest quality, importance is safety, because if somebody makes a product that's unsafe, that causes a problem for everybody. From my own experience in terms of, making a number of recombinant proteins, having precise criteria and specification around the product, but also having that specification confirmed by others, gives a level of reassurance and transparency, so that if somebody else looks at that product and says, well, actually your definition is okay, but it didn't do the right thing because you missed this certain quality attribute, then they could then say, well, it's worth still looking at this because we can change that quality attribute. I think it also gets easier with platform technologies. Obviously making batches of recombinant trimers using the same process, it's relatively easy to quality control than making one day a viral vector, one day a recombinant protein, another day something that's a viral-like particle. So, platform technologies will help. They're not the only solution. In the context of, for example, RNA vaccines, again, the release criteria can be very straightforward. And because they're genetic sequences, people can actually go and download that genetic sequence and try it in their own lab if they want to verify it. So I'm not concerned so much that we may make something inappropriately and it fails, because I think others will look at the same structure and model it themselves. I also think while having a pipeline to develop a product is of real importance, we have been in a siloed position in HIV of only thinking about how do we make a product when we've got one. And I really think we still don't understand how to make the concepts of how to make a protective HIV vaccine. And so if one actually focuses on understanding the rules, for example, to get broadly neutralizing antibodies, and you actually solve that problem, we may not be immediately ready to push that into a global appropriate manufacturing cycle. But having solved that problem would be such a huge step change that then solving the production issues, I think will be a much lower bar. And of course, the incentive to invest in that will be much higher. And then your question about groupthink, obviously, if it's top down, what gets tested is determined by the elite, then that will potentially lead to groupthink. But as long as there is space, and I think there is always space and opportunities for maverick ideas to be able to get into clinical trials appropriately sized. If they work, you'll suddenly see that this is something that's worth pursuing, and others will pick it up. And again, an example, we're doing a trial with a very novel immunogen in Europe with 8 participants. It probably is extremely unlikely to work, but there's enough science in there to say that if it does induce broadly neutralizing antibodies, it could be game-changing. And so I think there will always be room for those small scale, let's see if this creates a new paradigm in a clinical setting. But over to Carl.
So I'll reinforce a couple of points. I think the platform technology is going to be essential moving forward, as Robin mentioned, the messenger RNA. But I think some of the critical work that's being done right now to manufacture both the protein version, validate it to the extent possible you can ever validate a protein, and then compare it in terms of the immune response you see to a messenger RNA is an essential part of some of the work that is ongoing already. That kind of background information will be critical for us to really decide once and for all that mRNA can really do what it needs to be done. I think we have to also be aware that when we modify a trimer by putting unnatural cysteines in or something along those lines, it may not fold right if it's made from messenger RNA, and all of that needs to be worked through. Secondly, the whole issue of clonal response in terms of the groupthink. That is a problem and has been a problem in HIV vaccines. Let's acknowledge that up front. We've been through waves of first non-neutralizing antibodies, then CD8 responses, then back to non-neutralizing antibodies and maybe combining with a CD8 response. And it has yielded essentially nothing. And we really need to shake ourselves up and say, we're open to ideas. But this is where the small animal models with genetically engineered mice testing specific ideas can get you the data you need as a group with a novel concept to say that this idea is merit. Just because you have the idea and it's unique and different doesn't mean it's valid. You have to have data to support it. And I think that's where the space is needed, is enough research funding so that if somebody has a novel idea, they can either prove it and move on or get out of the business and move on to the next idea.
Thanks, Carl. Well, I think as everyone has, emphasized, at this stage of the HIV vaccine effort, experimental medicine studies are likely to play a critically important role. And as we think about the broader, response to the HIV pandemic, we all recognize that engagement of communities, often people living in resource-limited settings, is really the nature of the medical and public health need that we need to address, and we need to find ways of aligning our research approaches with the locations and populations where the need for an HIV vaccine is ultimately greatest. And you've heard from the different speakers so far the importance of ethics and community engagement and communication. And given the importance of those, I think it's really valuable for us to focus on those questions now, and we're fortunate to have a great group of panelists to do so. So perhaps I will turn it over to Christine, but perhaps she can go first, and then Maureen, if you want to have additional comments about how you see experimental medicine studies presenting potentially different ethical considerations or additional ethical considerations versus vaccine clinical research in general.
Great. Thank you very much for including me on this panel. The first thing I want to say is, because the issue of value has come up quite a bit, I want to say that value in studies is an ethical requirement. And it's an interesting ethical requirement because I think it's required in all research, whereas benefit to individual participants is not required in all research. And so being able to judge the value of these studies on a study-by-study basis, but also in the context, as Carl was describing, of the full scope and the end game, the value of the study to advance progress in HIV vaccine understanding or development is really quite critical. And it's critical at the front end in the sense of, making sure that only studies that will advance value are approved, but also in being able to explain to any review committees and communities and to invited participants what the value of the study is. And basically, the value is increased or enhanced, I guess, by disseminating and sharing the results widely and sharing data widely so that others can build on them and also use them, as was described already, communicating across different investigators and groups and other ways of making sure that the sort of context is put together. And certainly sharing results or what was learned from a study with the participants as a way to respect their contributions. But you asked, Mark, one more thing I'll say. You asked what's different here. So, certainly in experimental medicine trials, as I understand them, all the usual ethical considerations, the collaborative partnership and scientific and social value and rigorous design and fair participant selection, informed consent, all those things, they all apply. What's different here, I think, and several people already have alluded to it, there may be more of a chance of misunderstanding the value of the studies or the purpose of the studies or the lack of personal benefit that might accrue from any study or the limited or lack of personal benefit. And so there needs to be even more attention than usual, I would say, in engaging the relevant communities and the potential participants in understanding those things. So a clear explanation of those things in the process of informed consent is really essential. And then, of course, if informed consent is an important protection in terms of people understanding the limited amount of benefit, then we really also need to pay more attention to whether or not or when to engage groups who can't give their own consent or have less capacity to give their own consent than others. The only other thing I would say is maybe different is there's, when we talk about trials of vaccine products, we often talk about whether or not those products will be available to the local communities in which these studies are being done. And I think the question about what benefits to the local communities beyond the value of the knowledge, if any, should be considered is something that we might want to talk about. I don't know if Maureen has more to add.
Yeah, thanks for those great perspectives. Maureen, I'm sure you probably have additional thoughts to add as well.
Yeah, I'm happy to come in. And I think I just wanted to emphasize on the importance of communication. I think which a lot of panelists have really touched on to say, I think it's very important at the onset to make sure that the goals and objectives, right, of experimental studies are clearly communicated in a transparent manner, right? When I say transparent, I mean, all details must be shared, right? And this is the only way that we can gain public trust. I think as we'll be conducting this research, making sure that we are clearing all the myths and misconceptions. It must be very clear, of course, to the study participants that, they are being, of course, invited to participate in research, and that the objectives, right, of the research may not necessarily be based on their individual best interests and or preferences, which is, the case for experimental studies. We know, for example, if you take, for example, an experimental HIV vaccine trial, where participants will be randomized to an experimental HIV vaccine placebo, we know that both of them, may not provide benefits. So making sure that that is communicated to participants, and making sure that as participants are joining the research, they don't have misperception that they could be protected against HIV in the case, if you think of HIV vaccine. I think is very, very important. Now, talking about the issue of ethics, we know that scientific research, cannot effectively be conducted without necessary safeguards, but also the public trust. As such, I think the do no harm principle, right, should be at the core, should be at the center, or should guide the development and the implementation of experimental medicine studies. And I think just like with other research or studies, it is the responsibility of the research team to ensure that the study volunteers, are appropriately informed about the risks and benefits of a procedure, just like we do with other studies, but even more so important for experimental medicine, making sure that they understand. In that respect, for, I think, to a point that Christine lays, respect for individual autonomy to decide whether not to or to participate in the study, I think is very, very important, and that the decision to participate must be very voluntary. And the decision, of course, must be free from coercion, but also undue influence. It is important, of course, to ensure that the informed consent process, right? It's not just focusing on, protecting the investigators or the research institutions from liability that may come, right, with the enrolling participants on external medicine, but rather the focus should be making sure that the study participants are informed and they're able to make that informed decisions. And also, I think I'll just talk about like the consent process. I think this has been a concern sometimes when you talk to study participants, like through committee advisory board, sometimes the consent form can be too lengthy, too wordy, too complicated. Making sure that they are very concise and very specific and easy to understand, so that again, contributing the fact that participants must be able to make that consent response. And lastly, I think I'll just talk about the importance, of course, of partnering, right, with communities like community advisory boards, and even involving them in the designing of the consent form, so that they can be able to point to what are the critical nitty-gritties in terms of information that must be included in the consent form that respects the cultural values of a given community. So let me stop there for now.
Well, I actually don't want you to stop, because I have some additional questions for you. I mean, you raised the importance of participant understanding and the completeness of the consent, but also the clarity and conciseness of it. And I'm wondering, given that the questions being asked in the experimental medicine sphere and the technologies being used to address those questions are quite sophisticated these days, I'm wondering, do we need sort of new tools or new approaches to sort of engage communities with accurate information, whether it's in just broader awareness or around the specifics of people who are considering participating in this study under the umbrella of informed consent?
I think the traditional methods of creating awareness, community awareness, but also providing information to study participants, and think could work. But also, I think where because I think we shouldn't take the fact that, for example, if there are any scientific words that don't have like I'll take an example of my language, Chichewa, to say if you don't have a translated a Chichewa word for any specific scientific word, that shouldn't be an excuse, for research team to still explain to the study participants what that means, because it's important to make sure that all the complexities about the studies, about the candidates or the treatments or the platforms that are being used in the studies are being communicated. So maybe use of visual, right? Using visuals, right? Where, for example, written, for example, text, recruitment script, for example, may not provide all the detailed information. The use of visual, can also be very effective. But also, I guess, combining, there are studies that have been conducted, to test like which is the best, consent form process, whereby when you combine your written consent form and that verbal explanation and adding visuals, right, it also helps, to increase that comprehensiveness or the comprehending of the study itself. So I think using innovative ideas like, the visuals, but otherwise, by the end of the day, we need to make sure that, the information is communicated and that the participants, they fully understand and they're aware of what they're getting themselves into to avoid any frustrations or any harm by the end of the research. Thank you.
Thank you. Christine, did you have any thoughts you wanted to add?
I agree with Maureen, and I would just add that, there has been a lot of research on informed consent and methods of informed consent that might improve understanding in different contexts. There's a lot of examples of very creative use of analogies and visual and graphics as ways to explain things to people. Nonetheless, informed consent is not perfect anywhere. And so, it behooves people doing these trials to engage communities, as Maureen said, so that the community advisory boards and others can help, sort of advise on what kind of language, what kind of processes for transmitting information, and how to gauge whether people understand sufficiently. But it is really important for people to understand what these are about and why they're being done. The value of them is critical to communicate.
Thank you. Well, I want to encourage the participants to put questions in the Q&A as well. We have about 20 more minutes for this discussion. I have lots of questions that have come up that I could, fill the time, but we want to engage you as well. One question relates to regulatory strategies, and the other relates to where these studies are done, and some of them aspects are connected. So, Robin had highlighted, some differences in regulatory approaches in the UK versus the US, which are obviously important to understand and navigate. But in many ways, these types of studies may be new for regulatory authorities in countries where HIV incidence is high. And I'm wondering, what do we need to do to ensure engagement with regulators in those countries and foster regulatory science understanding and the tools necessary so that experimental medicine studies can actually be done in places where the HIV pandemic is having its greatest impact. I don't know, Carl or Christine or Jerome, if you want to, or others might want to address that question.
I'll go ahead and start. I think what we want to continue to do is to build core competency across communities. And by that, I mean that we support the education and training of bioethicists in countries where we are working so that there is a strong ethics community that can tackle these questions. We also work with the regulatory bodies in terms of how we can strengthen them, and as well as seek to train and teach and support strong CABs across our research. The NIH has a huge clinical trials infrastructure throughout the world, and CABs are critical. But some of the limitations we have are the CABs are very focused on a trial by trial basis. There's not a whole much else we can do with that, but through partnerships with academic institutions in countries, there's more that could be done. So I think we just need to continue to constantly keep this front of mind, that we need to make sure that we are training this generation, the next generation, and the generation behind in all of these areas.
I can't agree with that more. Training more bioethicists is always a good thing. I also think that, you know, in fora like this one, including bioethicists in the discussion is also a very helpful thing, because it's not always obvious what the ethical issues are, what the ethical requirements are. And as Carl said, strengthening the bioethics capacity on the ground in all the places around the world where studies will be done is also very important. And engaging the regulatory people to the extent that they're willing to be engaged in the in understanding the value of these studies and how they're different from other kinds of studies that they might be reviewing, and why they're important to do and how they are done with care.
Great, Robin, I see that you want to add something.
I mean, I think that the regulatory oversight of experimental medicine trials, particularly at a global level, is a mess, to be frank. And you know, I think we're all aware that many countries that have a less mature regulatory body or experience often look to the FDA for a lead. And so I think that, you know, by doing appropriate experimental medicine trials as exemplars in the US, and showing what can be done under FDA you know, legislation, will inform the way experimental trials are done in other parts of the world. And it's complex. You know, I've looked into it. I've spoken to a number of regulators recently. It's complex in Europe. It's down to, you know, individual national governments' interpretation of European legislation, and you know, that it's interpreted differently by different regulatory bodies. So we did 2 trials that were identical in different parts of Europe. One had to be done as a traditional phase 1 trial with all the bells and whistles, and one was done as an experimental medicine trial with a much reduced safety requirement in terms of the information that needed to be recorded. That's not saying that it was less safe. And so there's also probably a good space for something like an opinion piece to be published, highlighting the issues and making some recommendations to harmonize approaches at a global level.
Thanks, Robin.
May I add?
Sure, please, Maureen.
Yeah, just quickly. Again, just to say that, you know, the role of the local regulatory bodies, the ethics committee, I think cannot be overemphasized here. And I think we know that most of these experimental studies, you know, they happen in high income countries. Maybe it's high time, I think we also start considering how can we also move, right, you know, shift, you know, or ensure that low income countries are also participating in such kind of, you know, experimental studies. And by doing that, I think it provides an opportunity to build capacity and build the capacity of researchers, build capacity of the ethics or the regulatory bodies, but also it enhances, I think, that community research literacy for communities to have this knowledge, you know, this new knowledge, you know, or novel knowledge, you know, around experimental studies. So making sure that the ethics, you know, research committees or the regulatory bodies, you know, have the capacity. It could either be through training, right? It could either be through matching, for example, with you know, matching members of the regulatory bodies with members of regulatory bodies in another country, or in you know, from matching regulatory members from low-income countries with the regulatory members, you know, for example, from, let's say, from the US or from the UK, right? So that there's that continuous mentorship, you know, but also, you know, we're providing that continuous capacity building, you know, to the members within the research. So yeah, the role of the Ethics Research Committee cannot be overemphasized. And I think a lot of the issues that we talked around ethics actually starts with the Ethics Research Committee. If the Ethics Research Committee is weak, then, you know, the whole trial is going to be a mess, right? In terms of ensuring that the informed consent process is documented, is clear, and so we need to make sure that we tighten the board at the Ethics Research Committee and that they have the capacity to control, but also regulate the experimental trials. Yeah, I wanted to add that. Thank
you very much. There's a question from the audience that I know a number of people on the panel have, you know, previously discussed, from Kundai Chinyenzi. It's really related to as Robin had articulated, experimental medicine studies in most, if not all instances, will have a lack of direct benefit to the participants, but the participants are being asked to engage in often detailed studies that involve specific procedures which are outside of the realm of a traditional vaccine trial, such as fine needle aspirates or leukapheresis or things like that. And given both the combination of the lack of direct benefit as well as the more intensive commitment that's being asked of the study subjects, the study volunteers, is it appropriate to consider different models to incentivize trial participation? And I think a number of people, including Christine and Carl and Maureen and others, may want to comment on that question.
Or I could start. I mean, I think, as everybody knows, it is sometimes challenging to recruit people in any kind of study, and they need some motivation for participating. And oftentimes people are motivated by their interest in contributing to science, but also sometimes motivated by potential benefit. And so, as you just said, Mark, the reality that these studies have very limited amount of benefit might change the motivation structure. But incentives can be used for lots of things. They can be used to motivate people who otherwise wouldn't participate and therefore enhance recruitment. They also can be used to enable people to participate who wouldn't be able to otherwise because they couldn't afford the time or the transportation or the things that they're giving up in the time that they spend at the study site. And a third reason is to show respect for the participants and their contributions and the amount of time that they and inconvenience that they put into participating in a study. So there are lots of good reasons to provide incentives in a study. And studies like this might very logically include incentives. Those can be cash, but they don't have to be cash. They could be other forms of incentives, food or treatment for something else, or other kinds of incentives that people might find appealing. And I think some of the basics, like helping with transportation and maybe meals while they're at the site or something like that, should be sort of standard. They should be considered necessary, not added incentives. The thing that people worry about with incentives is that they may be so attractive that people will not pay attention to the risks or not understand that there is limited or no personal benefit to be had in the study. I think this is what people call undue inducement, and it is a concern in some cases. I think the evidence that exists from studies that have been done on this topic suggests that it's less of a concern than people worry about. In other words, people actually do pay attention to the risks. They do pay attention to what they're being asked to do. And sometimes when money is involved, they might even pay more attention because they're like, this is different, So I don't think it's a reason to avoid incentives in studies like these. What incentives to use, I think, should be up to the community. The community values and preferences can help shape what type of incentive, amounts of incentive, et cetera, for studies of this nature.
Thank you. Maureen, Carl, others, I don't know if you want to add to that.
Oh, I mean, I don't think I have much to add, but I think just to agree with Christine that incentives are really important. And I think whilst we are talking about benefit, I think it's also, as we're having this discussion, it's also important to acknowledge that, no participants in an HIV vaccine trial has received a benefit of an effective product. So I think it's so important for us to have that at the back of mind. Just to emphasize that I think as we're thinking of incentives, I think it's important to make sure that it doesn't take away that autonomy, that freedom for someone to decide whether they want to participate. I think, yeah. So I think
We're losing your connection, Maureen.
Yeah, Maureen is frozen up. I think one thing that we could do as researchers is that if the purpose of an experimental medicine trial is to test a specific hypothesis, be able to accurately and clearly articulate which procedures we're asking people to undergo will address that hypothesis, and really avoid the addition of add-on experiments for the sake of researcher interest, and really keep it a clean, simple, straightforward experiment to the extent possible, so that we don't have one key hypothesis and 100 exploratory ideas where we're drawing bloods just for the sake of exploration. I think that that's where we're going to get into trouble. And so if there's a clear line between this procedure and the answer, and that this is part of the informed consent, I think the clarity of that will be essential. It also keeps the thinking clear on the purpose of the experimental medicine trial.
Thanks very much, Carl. The time is, fleeting away, and I want to make sure that we can capture any closing thoughts from the panel members before this session comes to an end. And maybe I will start with Jerome to see if you have any additional thoughts. Are there any things you want to emphasize? Were there any things missed in the discussion you might want to flag for future dialogue?
Yeah, so I think that we covered, the critical points. I mean, we came down to to doing this, to starting to do experimental medicine studies in the global target populations, which I think is really critical. And to the extent that requires strengthening of participatory practices, ethical practices, and regulatory regimes in different countries around the world, I think that's all going to be a good thing. And again, I keep coming back to, I think that this experimental medicine approach is important because, we've had not a history of success in HIV vaccines. And in the end, I think most of us believe that it's the science that will win out in the end and that will bring us to a solution. But I really have to emphasize the need to think through the process and the product as something that goes with the experimental medicine approach, because we have been frustrated with other vaccines that haven't shown the same promise in extended human clinical trials as they did in animal studies, where we very clearly understood what we needed, the immune responses that we needed to achieve. So think through the necessary parts of quality of the experimental product used in experimental medicine studies. And just having a platform does not necessarily mean that you can transition from, say, a pox vector made in eggs to a pox vector made in wave bags, because it can be a very different product in the end.
Thank you. Maureen, any closing thoughts?
Yeah, I think I would just emphasize that I think it's important that we bring communities into this conversation around our paradigm shift. I feel like, communities have not been part of this conversation. And we know that communities can only engage and support any new trial design only if they are brought along. So I think making sure that communities are part of this conversation, I think is really critical. And that's actually part of the work that we are doing at AVAC around the Trial Design Academy. So I think making sure that communities are brought along the conversation, I think would be very critical moving forward. Yeah.
Thank you. Carl, obviously you've thought a lot about this, and I know you know how multifaceted it is. Is there anything you want to emphasize or something you think we've missed?
I want to leave this group with 3 thoughts: quality, community, and focus. Quality of the questions, quality of the product, quality of informed consent, engagement in community, and then once again, hyper focus on the specific question that we're addressing in the experimental medicine trial.
That's great. Thank you. Christine, obviously this is a very important but also very complicated area, and you've highlighted a lot of the key elements of it. I don't know if there are any that you want to reemphasize or add.
Just to reemphasize the importance of critical evaluation of the value of the study before it starts as it's being communicated and reviewed, and when it's finished, so that it can be coordinated with other studies and add to the value.
Great, thank you very much. And we will be ending where we started with Professor Shattock. Obviously, I know you've thought deeply about these issues as well and are actively engaged in them. I'm wondering how you would like to reflect on today's discussions and what more needs to be done to move this dialogue and the implementation of this vision forward.
I mean, I hope moving forward from today, we can, anticipate a step change in the way we focus on these issues. I look back in my notes, and the first time I talked about experimental medicine trials for HIV vaccine research was in 2012. So we're a decade from first discussing this. We're in a different place in our understanding of what we need to solve for HIV vaccines, and we have new tools. But I hope we won't be having this discussion in another decade's time, but we'll be looking back at a moment where we changed the paradigm and we moved into a new era of vaccine research.
No, the timing you mentioned is interesting because maybe we didn't necessarily have the hypotheses or even the tools to be specific about experimental medicine studies, but those of us who have been involved in the HIV response for a long time, appreciate how critical a role that what you might now call experimental medicine studies played in our understanding of how to both develop and deploy effective antiretroviral therapy and our understanding of AIDS pathogenesis. And clearly that insight dates back decades. And in many ways, the things that were done in the 80s, 90s, really laid a foundation demonstrating the value of this approach. And hopefully based on, the feedback that you've all provided and the promise that you've articulated and the sort of, I think, thoughtful guidance about how we can approach this in a responsible, focused way around, as Carl indicated, doing this around the right questions, using the right tools in the right way, and obviously sharing information and insights, and maybe using this as an opportunity to really weave together a number of really very promising threads in the HIV vaccine field. My experience is that there's tremendous willingness to collaborate. The collaboration and sharing state-of-the-art technologies is going to be essential for success here. So we'll do best in this arena if we do it together around a thoughtful approach. And I think that there's a very important dialogue to build upon, what Carl had talked about before, about how we think about a more, better communicated, better aligned approach to answer the key questions and try to expedite the development of an HIV vaccine as quickly as possible. So I just want to thank all the great panelists so much for their very thoughtful remarks. And at this point, we're unfortunately out of time, but I can turn it over to Linda Gale Becker, whose, I'm sure, perspectives we all will both value and be stimulated and cheered on by. So Linda Gale, back to you.
Thanks, Mark, and really expert moderation there. I think we covered a really wide-ranging set of thoughts and ideas. I think we sort of, to a certain extent, raised some controversial aspects, and I really look forward to the next webinar that is going to take us forward in this discussion. But I do want to reflect for a moment. I too was thinking about, from whence we have come in terms of treatment, and just the sort of winding road, if you like, that taught us so much about this virus that we know today, that the iterative nature of the work that brought us to this point, and just, the excitement of really embracing a new paradigm. What I would wish for us all is that we truly would take up some of the ideas that have been raised today. I couldn't agree more with Carl around the focus, around the need for a reductionist kind of approach, just the wonderful thoughts around bringing community along and remembering that community involves ethicists, regulators, communities of scientists, communities of advocates, communities of participants. All of these people need to be brought along as we start this paradigm. And I would just say that may we do it with that same passion and zeal that we undertook treatment, but with the enthusiasm and urgency with which we've seen the more recent COVID experience, and apply some of that to the next era. I take this moment, I realize we're in the last few seconds, to just really thank Jerome, who's on the east side of the globe, and it's late for him. I want to thank Carl, of course, Christine, who's on the more west side of the globe, and you, Mark, for getting up early this morning. Thanks to Robin and Maureen and myself sitting here in the middle, and all of you for participating in this era of virtual webinars. I know it's early for some, late for some, but it really does create a solidarity around a problem we all want to see move forward as quickly and as efficiently as we possibly can. So at this point, I'm going to thank everyone. Thank you, Robin, for creating a talk, each one of you for giving your insights and wonderful inputs to this. And I'm going to hand back to Elena as she sets us up for the next webinar. I look forward to seeing you all at the next webinar. And thanks again to everyone.
Thanks, Elena. Thanks to you all for your time today. Our second webinar, that will be the last one for now on this series, will take place on the 1st of February and will be on experimental medicine trials for HIV vaccines research. You can register already through the MailChimp or in our Global HIV Vaccine Enterprise webpage. Finally, just to ask you to fill in the survey that you can get it at the end immediately when you close the Zoom session. You will get it in your browser, or you can also fill in through the MailChimp invite when you get it. Thanks once again, and hope you can join us again soon. Nice day.